All right. I'd like to thank the organizers for inviting me. I've been enjoying this workshop quite a bit. I'm going to tell you today about some work that I've done in collaboration with some colleagues at the University of Washington, who I'll introduce to you in a bit. A lot of this work came out of my participation in the ENCODE consortium, which I've been involved in sort of peripherally since its inception back in 2003. And one of the frequent pieces of feedback that we get from the scientific community is that they're not happy with our selection of which particular cell types we chose to do the analysis on. This is, an understandable frustration. The process of deciding which kinds of assays to do in which cell types was necessarily a little bit of a Wild West. In the beginning of ENCODE, many of these technologies didn't even exist. In the beginning of ENCODE, everything was chip-chip, and it became clear eventually that maybe we should be doing chip-seq instead, for example. And a lot of the decisions were made based on practical considerations like, can we get cells in enough quantity to distribute them to lots of different labs, and so on. But as we grow the amount of data that ENCODE has produced, it becomes clear that we would like a more systematic way to make these kinds of decisions. So this is an annotation, sort of a schematic that shows the available data in the ENCODE data matrix at this point. On this axis, you have cell types. These can be, you can think of them as sort of tissues and so on. A lot of them are immortalized cell lines, but they're also primary tissues as well. And then along this axis, there are assay types, some things like chromatin accessibility assays like DNase 1. Of course, all the kinds of histone modification assays that we talked about for ChIP-seq, and then many, many transcription factor ChIP-seq assays as well. And the black dots up here are all of the ones that exist, and all of the white is stuff that hasn't been done yet. So as of, I think, yesterday, I checked, and there were 3,373 data sets that had been done in human. And you can see that this makes a very small proportion of the matrix of possible experiments that one could do across all of these cell types. And of course, the axes here go on, right? There's tons and tons of additional types of cells and perturbations and experimental conditions and so on that you could imagine assaying. And of course, many other transcription factors, for example, that have antibodies that we haven't done any experiments on. And also I wanted to make the point that as this data has evolved, one of the things that ENCODE has been trying to do is develop standards for analyzing this data. I think Eric pointed out rightly that initially, like in phase 2 of ENCODE, there really wasn't much in the way of standardization because the technologies were quite new. At this point, all of these data are actually available from the encodeproject.org website. They've all been uniformly processed by the Data Coordination Center, so that problem at least has been addressed. Okay, so the question that we face moving forward, and this is a question not just for ENCODE, but for scientists in lots of different situations, is if we want to decide on doing some new experiments, we presumably have a limited amount of cash, and we want to get the most bang for the buck, right? So the question is, if the particular question that I want to try to address is, let's say that we're disappointed that our cell type wasn't studied at all, and we're going to do a small number of assays to try to get an idea of what's going on in that cell type. And we would like to choose the ones that are going to be sort of complementary to each other. So we'll think of it as like a panel of assays with some cost associated with it, and we want to choose that panel so that it's the most informative. For the purposes of this talk, I'm going to assume that all the assays are equal cost. It would be straightforward to generalize to putting in the cost as a sort of a weighting factor. So you can think of this as a specific example of a more general class of problems where you have some large collection of data, and you want to choose a subset of that data that tries to retain the statistical properties of the full data set. So it's a sort of a representative subset selection. You can think of this large collection of blue dots, and the red dots might be the representatives that are selected to represent it. So this problem, the one I just described, falls into this setting. But you could also imagine, for example, if we wanted to train some expensive machine learning method on the ENCODE data. Maybe you don't want to train on all 3,000 assays because it's a gargantuan amount of data. You'd like to choose a subset that is representative of the Fuller set for training a computationally expensive model. Or maybe even just to try to make sense out of the data. You'd rather not visually look at all of the data, so you want a subset of existing data. There's also a similar problem that I'm going to talk about later in the talk where that comes up a lot in protein bioinformatics, where you have a set of protein sequences and you'd like to choose a representative subset, say, for example, for building homology models. And these all are examples of this general problem. Okay, so the structure of the talk is first I'm going to tell you about an optimization framework for this representative set selection problem, and then to talk first about choosing panels of genomics assays, and then a little bit about the protein set selection as well. And I should say that this is joint work with my longtime collaborator Jeff Bilms in electrical engineering, a graduate student in my lab, Max Liebrecht, and a graduate student in Jeff's lab, Kai Wei. Yeah, I'll talk more later about what the different roles are. Okay, so let's start out with an SAT question. Those of you who took the SAT remember these kind of analogies, right? So you have two things that are similar to each other, and then you have to choose the other thing that's similar. So continuous optimization is to convexity as discrete optimization is to, and there are four choices: semi-definiteness, submodularity, differentiability, and perspicacity. So everybody who thinks that discrete optimization is similar to, that the relationship between continuous optimization and convexity is the same as discrete optimization to semi-definiteness, raise your hand. Got to choose one. All right, we got a few. Submodularity. Okay. Differentiability. And a lot of people apparently are going to vote for perspicacity.
Because I have no idea what it is.
The answer, which got the most votes, is submodularity. So this is the... Sorry?
I'm kidding.
So I will explain to you what submodularity is if you're not familiar with it. Sorry?
What perspicacity?
Perspicacity is totally unrelated to mathematics. So. Yeah, right. So let me define what submodularity is, and you can all Google perspicacity while I'm doing it. So first, before I can define what a submodular function is, I have to define what a set function is. A set function is a function that takes a set, usually called a ground set here, V, and it assigns a value to every possible subset of that ground set. So this function f here is saying if I select these guys that are circled in red, then we get some value, in this case 3.2. So this is the set function, and for the purposes of this talk, you can think of this number as being related to the quality of the subset. Okay, so one immediate problem that is apparent is that if you wanted to try to maximize this particular value, the 3.2, how long do you think it would take using a naive algorithm to find the best possible subset? 2 to the n, right? There's 2 to the n different possible subsets. So that's a long time, typically, unless your subset is very small. Okay, so a submodular function is one that allows us to optimize this more efficiently than 2 to the n. And to define submodularity, it's the basic idea is it's the law of diminishing returns. So in this example, you have a set A that's been unioned with a singleton set that has this one example in it. And then you look at the difference in sort of the quality or the function value between that set and the initial set A. And this term you can think of as the gain that you get from adding this one additional guy to the set A. Right? And here is the gain of adding this guy to a larger set, A union B. And so the law of diminishing returns just says that this gain, when you're adding to a smaller set, is larger than the gain that you get when the set that you're adding to is large. Okay, so as you have more things, new stuff becomes less valuable to you. This you can think of, if you can't keep that straight in your head, you can think of it in terms of McDonald's Happy Meals. So imagine that you're at McDonald's and you want to get some fries. If you just have a burger, you want to get a drink, and if you just have a burger, the cost of adding a drink is larger than if you buy fries, because the fries make the additive cost of the drink less. So Happy Meal pricing at McDonald's is an example of submodularity. Or diminishing return, yes.
Increasing weight.
Yeah, and increasing weight as you go. Okay, so a nice feature of this general class of functions is that you can define a relatively simple greedy algorithm where you essentially take an empty set and then consider each possible member of your ground set to add to the empty set and choose the one that gives the most boost to the quality of the set that you've got so far. And this, interestingly, is guaranteed to produce a solution that's within a constant factor of the best possible solution if you did the full exponential enumeration of all possible subsets. So the factor is within 1 minus 1 over e, or a factor of 0.63. Interestingly, it's actually the case that this is the best you could possibly do unless p is equal to np, which is generally considered not to be likely. And then finally, it's also the case that there are some fast versions of this algorithm that in a practical sense run at about k log n. So it's quite quick to be able to do this for large data sets. So this kind of thing is sort of the key result that drives a lot of this work. And in fact, part of the reason we got started on this project was that my collaborator, Jeff Bilms, has been applying submodularity very successfully in a variety of machine learning applications. So submodular optimization has been used in many fields in economics and circuit design, and there's been a special issue of discrete optimization journals devoted to it, but it hadn't really been used much in machine learning until the past maybe 8 years or so. And it's shown a lot of success in summarizing things like large corpora of images. You want to find a representative set of images, or even natural language processing. You take a text, choose a subset of sentences that capture the sort of the gestalt of the text. So Jeff sort of had this hammer, and we were looking for a nail, and then ENCODE had this problem, and they fit, very nicely together. Okay, so I'm going to show you how. Yeah, question.
This is true in the limit of large n, or this is a correct statement for any?
Good question. In the limit,
that blew my mind.
It's got to be in the limit of large n, right? Wait.
I think it's true for every instance.
Wonderful. Okay.
We'll have to find out.
By what measure? Anyway, that's like a.
But the number that you're optimizing, right? The value that you get and the value of the best possible have a difference that is no larger than this ratio. Yeah. Okay. Okay. So, are you okay with that? You're choosing a representative subset according to some submodular function, and you say that the quality, the score of this representative subset is within a factor of 0.63 of.
I redefined f as the square of the old f, and then I don't understand why it's 1 over e.
Well, so there's different f's. I mean, it's for any given f, right? So as long as f is submodular, then this will be true. Okay. Oh, and I should also mention, intuitively, I didn't say this earlier, but you can think of submodularity as kind of a representation of information in the set. So entropy is an example, a canonical example of a submodular function, right? The submodular is much more broad, but entropy is one common function that is submodular. Okay, so let's talk about this particular problem. So we're trying to decide what panel of genomics assays should we perform on a new cell type. So imagine I've drawn this now as sort of a tensor. Now you've got genomic position going back this way. Here's all the experiments that we've done. We're getting some new cell type. And what we're going to do is first compute a matrix of assay similarities, and then give some budget. So maybe in this case, the budget is we can afford to do 3 assays, have a selection procedure, and then get out our panel. Okay? So for the purposes of these results, we've focused just on using correlation as our measure of similarity. This is definitely an area where you could imagine having other similarity measures. We didn't vary that particular thing, partly because there was plenty of examples in the literature of people using correlation in this way, like what Martin did a couple days ago. He showed a heat map very similar to this, where you were looking at correlations. There could be lots of interesting directions to go varying how those similarities are computed. But for now, we just focused on saying 2 assays are similar in the average correlation in the cell types where they've both been performed. Okay. Now, the challenge often in doing this kind of optimization is deciding on your submodular optimization function, right? So we actually tried a variety of functions in this context. In the second part of the talk, I'll tell you more about different kinds of functions. But for now, I'm just going to focus on the one that we decided upon that worked well, and it's called facility location. This is a well-studied function. A canonical example that people use is you imagine that you are Nike and you have a number of stores in and around Paris, and you want to choose the location of your next Nike facility. How do you do that? Well, one heuristic you could imagine is saying if I sort of average over all the places where people live in and around Paris, you'd like everyone to live pretty close to one Nike store. So you sort of think of it as sum over all the people, the distance from that person to the nearest Nike facility, and that just is the facility location function. So you're taking, in this case, the sum of similarities to the most similar representative. This is meant to represent the distances from this guy to the Nike store and so on. So you're adding up all of the lengths of those red lines or the similarities, and that becomes the facility location function. So those are the two things that we use to do this calculation. We take these correlations, we use those as our similarity function, and then we can compute for any given set of assays the total similarity among them, among all the other assays in the ground set to the assays in our panel. Okay, so the question is, how do we know if it worked? You can start by saying, so we did this, for example, for a bunch of histone modification assays. These are just a browser shot representation of different histone modifications. So this is like histone 3, lysine 27 acetylation, and all these different codes stand for different modifications. And sort of, you can see I've marked the ones in black that are the first 5 that were selected by the submodular, the greedy algorithm. And, you can sort of wave your hands and say things like, oh, they do show sort of qualitatively different characteristics. Some have broader shapes and some are more punctate. But just visually, obviously, it's not going to be very easy to say whether this is a good panel in any absolute sense. So one thing that we did was go and look at how a panel of experts had, what a panel of experts had decided. So here is the choice order for our submodular optimization procedure. If we look at what the people in Roadmap decided to choose, they actually chose these 5. So 5 out of the first 6 that were selected by this procedure. So that's at least encouraging. Also not proof, but it seems like there's at least some agreement there. Before I go into the systematic measurement of how good this is, I think it's worthwhile to think a little bit about how we make sense out of this ranking. So this is that same list in rank order, and now I'm showing next to each one 3 different numbers. One of them is just the submodular score of this particular assay by itself. If you selected it first with no others, that's this modular value here, right? This is the conditional gain, which is if we add this to the previous one, how much additional value do we get? And then this is just the running total here, right? So one thing that's unusual about this list, well, actually, maybe I can ask you, what's one thing that anyone who's familiar, intimate with these histone mods that makes you unhappy about this list? Anyone who particularly, yeah?
The K4 monomethylations are...
Oh, that wasn't what it was supposed to be. That might also, what's wrong with the K4 mono?
I'd like the H3K4 dye to be maybe between the mono and the tri, as it's an in-between modification. I would have expected.
There's nothing
I see. Interesting. Okay, that's the one that I was expecting, the one about enhancers. But that might also, I'll have to look. I haven't looked into that question particularly.
Who's a surrogate for both, so I thought, well, I've seen that published.
Right.
But this may be pointing to the fact that correlation might not be, because see, you find the enhancer because often when you have di or trimetal, you also have some monometal.
Mm-hmm.
When you have monometal, you don't have trimetal, then it's informative. For
Right. So there's this
enhancer, right? And your score may sort of not be sort of sensitive to those kind of
these correlations. Yes, that's right. So in particular, a lot of people like to use this, the very last one here, as a very strong mark for enhancers. And interestingly, it's the only one with a conditional gain of 0. So at least in this setting, it's saying if you've got these 10, there's no reason to even bother doing the 11th. It doesn't give you any more. It doesn't get you closer to anyone in Paris, right? But the nice thing is that we can adapt this protocol to say, let's imagine that somebody has already done H3K27AC, or they really definitely want to do H3K27AC, and now let's just do the optimization from there on. So we say, this one is fixed, and now what should we do next, given that we've already done that one? And so you start seeing that, on the one hand, the conditional gain from this is smaller than quite a variety, several of the other, or sorry, not the conditional, the modular value of this one is smaller than several of the other assays. So that's why it wasn't selected first. But also, as you go down this list, you're consistently a little bit lower in the total quality of the panel than you are if you hadn't started with H3K27AC. So at least according to this metric, if the corresponding values here, it seems like putting this guy up at the top of the list is not necessarily a good idea. Yeah.
So I'm just a little confused. What is your distance between the nice one and the nice one? What is your metric?
Well, so it's a confusing analogy because I was using distance, but we're using similarity. But it's, imagine like 1 over the correlation or something like that to fit it into the Nike analogy. But it's correlation.
Across the genome or something.
That's right, across the genome. ZPing.
I think the difference you are seeing here is because all these assays are very gene-centric. So like the left panel is so gene-centric, and then enhancers occupy a smaller genomic real estate than
Right.
genes.
Right. So one of the things, when I gave this talk a few weeks ago, and we're computing these correlations is computationally kind of slow, so we haven't finished it yet. Kai said it was not quite done when I emailed him 2 nights ago. But one of the things that we'd like to do is try this again with correlations that are computed on a subset of the genome that is sort of biologically more relevant to what we're interested in. So that could be, what we're doing right now is DHSs, but you could imagine doing it on, genic regions or something like that. Yeah.
Does it make sense to include in the correlations like another functional output of the system? So for example, gene expression, because for example, H3K27 acetyl, maybe it's correlated with everything. It's both with enhancers and promoters. But what it tells is the difference in activity.
So the question is, could you imagine incorporating something like gene expression? I think you would have to build that, as far as I can see, into the similarity measure somehow. You could imagine some kind of weighted correlation, I guess. I'm not sure. It's not immediately obvious that you could fit that into the submodular function per se, but if you could find a way to put it into the similarity, perhaps. Okay, so, oh yeah.
In this setting, you already know the value, so you could compute the correlation, but you are asking about the new assay for which you have no information.
Right, so we're using so in this setting, we're assuming that the assay has been done in some other cell type, and we're using the correlations in other cell types in which it's been done. If you have never done the assay before, then there's no way you could know how much information it will give you. Yeah.
How influenced are you by distribution of the data you already have? So for example, if there's one that you've done on every single cell type, its correlation might not be, you might have a lot more variety, as opposed to one that's done on a few that are all very similar and they have similar.
Right, remember that. So the question is, the correlation that we're computing is an average over all the cell types in which the 2 assays have been done. And if one assay has been done many times, it's a much larger average. So presumably a smaller variance in whatever it's estimating. You're right, that could be an issue. I don't see it doesn't seem like you want to ignore when there are lots of assays to compute the correlation from, but I don't have any good solution for that problem. It is something that has come up before. Let me just go on, and we can have more questions later. I'll just say briefly that we also can do this, for example, for transcription factors. What do we? Already? Okay. Well, we might not do the second half, but that's okay. The second half was, Okay. So we also do this for transcription factors. You can see, for example, most of the transcription factors that we do in ENCODE are standard, transcription factors that bind at promoters, which is what these first three are here. But then you also have, for example, things involved in chromatin conformation, and then one of them that's RNA polymerase initiation complex. And also these guys are quite different from each other. But all of this begs the question, how can we sort of objectively evaluate whether this is a good summary? This was something that we had to wrestle with quite a bit in this project. And what we ended up doing was saying, well, let's think about how one might use one of these representative panels and then try to evaluate it in that context. So, for example, you might try to use a panel to impute data for the data that's not in your representative set. You might try to use it to identify functional elements, promoters and enhancers, and so on. And you might also try to use it for semi-automated genome annotation, like ChromeHMM or Segway, these kind of things. So we designed a sort of general train test cross-validation scheme where we would take all of the available data. We'd look at an evaluating cell type and all of the assays that are available in that cell type, and we would split them up. We partition them into what we call a source set and a target set. We do the assay panel selection on the source set, and then we do an evaluation to compare the selected panel to the held out target set and get some quality measure. So for each of those three scenarios, we replace this box with a different operation. So in the first scenario, that evaluation box, we're splitting the genome into a train and test set. We train a regression model, in this case a support vector regressor, do a prediction and just see what's the R-squared between those two. In the functional element prediction, it's very similar, except now we're taking an annotation. In this case, it's things like ChIP-seq peaks for known factors and just looking at the area under the precision recall curve. And then for semi-automated genome annotation, we take we partition the genome and assign it labels, and then ask how much variance does this annotation explain in the held out data. Okay. And the first thing we did was just look and see to what extent the facility location function agrees with these various external evaluation metrics. So each of these vertical axes are one of those three things. This is the R-squared, the area under the a precision recall curve, and the variance explained. And all of the blue dots here are just randomly selected subsets. We also included here is just sort of one heuristic for choosing a subset, which is what is the panel of assays that was most frequently performed across all the cell types. And then here's what was selected by the submodular procedure. If we do this more systematically, this is just many different random selections, and here's the submodular optimization. Again, I put the most frequently performed assay in here. And now we're doing it for different panel sizes. And so you can see that in all of these situations, the selection does a good job of optimizing this thing. That was just for one cell type. In the manuscript, we show it on a variety of different cell types. And then we also showed that you could generalize this to the setting where we're choosing among existing assays. As opposed to an entirely new cell type, we want to choose things from within a set of existing assays. And I'll tell you that worked well as well, because I don't have that much time. And then, like a minute or 2 minutes? Let me just briefly say, and this is in actually, I think this is on this is on bioRxiv. I'm not going to have time to go through the details, but one of the things that we struggled with in that first half was how do you objectively evaluate how this works. And one of my motivations for showing you the protein summarization task was that in that setting, we really do have an orthogonal gold standard, which is the protein structure. So this version of the problem is just something that people have been doing since the mid-90s. You want to find a non-redundant set of proteins that you're going to use for building a model of one kind or another, or doing any kind of analysis. These are the methods that people use. CD-HIT, I think, is the most popular. There's about 4,000 citations in Google Scholar, but I think that way undercounts how often these kind of methods are used, because they're just a part of many bioinformatics pipelines. And so in this second half, which you're not going to hear, we go through and compare we do summarizations on the SCOP gold standard, the SCOP set of sequences, and then ask how well our summary covers all of the different superfamilies in SCOP according to the structure. And in that case, we're able to show, according to this independent gold standard, that we do much better than the existing published methods. And maybe I'll stop there and take more questions. Thank you. Yeah.
Thank you very much. I mean, when you defined your problem, it's a very general optimization problem. And as you said, it's a hammer, and a very general hammer. The function, the facility selection function, also is not driven by any biological motivation. For you, the reason why you would want to look at the data only came in at the end in the application, the validation setting. And that's maybe how ENCODE works, but if I want to explore my particular cell type, I might have a biological idea from the beginning. So can you think of ways to change your, whatever it was called, this F function, to be more biological?
Yeah, so actually this is something that I had on my notes on one of the slides and forgot to say. So thank you for the question. The thing that we're doing right now, there are established methods for learning the submodular function. So you can parameterize a space, not all possible submodular functions, but a subset of submodular functions that are parameterized, and then you can learn the function itself, and that's what we're going to do. We're focusing on doing it in the protein sequence case because then we really have this orthogonal gold standard, but that's clearly where a lot of the value lies, and I should have pointed that out earlier. Other questions?
I can think of one more application.
Okay.
You have a
Oh, yeah.
very large set of genomes, and they have various distance, evolutionary distance, and you want to pick a subset for any application.
So we've thought of doing this. I was not thinking of evolutionary, but I was thinking in human populations. If you wanted to, imagine trying to test some kind of clinical treatment, and you've got a population of 10,000 people, but you can only afford to test it in 1,000. Which individuals should you choose? You could look at their genetic diversity and try to choose a panel in that way. And yes, this would directly apply to that.
Is there some way of adaptively choosing what experiments to do next if you're doing this iteratively?
Adaptive, yes, certainly. So remember I showed how you could put in H3K27 and assume you'd done that one, right? If you wanted to, you could run your greedy algorithm, find out who's next, then put it in and recompute your correlations based on that, right? And then do the next one, so that you could make it adaptive in that sense.
Am I wrong in remembering that submodular optimization can actually be done in polynomial time, assuming for any subset you know its value, which seems to be the same assumption for the greedy algorithm?
Assuming for any subset, I'm not sure what you mean.
Well, in the greedy algorithm, don't you need to know for a particular subset what its value is? Because
right.
Adding in one more to... Okay. I think relative to that...
I mean, you have to be able to evaluate, that's what the submodular function computes, right? So you have to be able to compute the submodular function on any given subset.
Yeah, right. But then there's a polynomial time algorithm that actually, there's an algorithm that only looks at a polynomial number of subsets in order to find the optimal.
But not the true optimal, just within the 1 minus 1 over e bound.
No. That's what I'm asking.
I don't believe that's right, but...
Minimizing a submodular function can be done in polynomial time. Maximizing...
Oh, is that what you're thinking? You're thinking of minimizing? That could be it. Yeah, that's true.
Just change the sign.
It's all right.
Other questions? All right. Thank you.
Thank you.